So we'll discuss the entire process of bilirubin conjugation. The bilirubin is end product of the hemoglobin metabolism. With bilirubin conjugation known as esterification. The bilirubin conjugation are actually start when the ancient RBC and will be destroyed by reticular endothelium system, which compromise the spleen, liver, and bone marrow. After the destruction of senescent RBC, there will be a formation of hemoglobin. This hemoglobin will be divided into there will be formation of hemoglobin. This hemoglobin will be divided into two fragments, the heme and the globin. The globin will be degraded. Its other product is amino acid, which will remain or stored in the amino acid pool. The heme will also undergo degradation. It will become ferrous iron, protoporphyrin. In this case, iron will be reused and will be transported. Yes, and it will transported in the bone marrow, whereas protoporphyrin will be acted by enzyme called heme oxygenase. When these two react, it will become biliverdin. Heme oxygenase will still act on biliverdin, and will convert it to an unconjugated bilirubin. Therefore, this is a water-insoluble bilirubin. The next thing that will happen is conjugated bilirubin will be transported to the liver to perform conjugated. Before that, B1 must bond to albumin and the specific site of conjugation of our bilirubin is in a rough endoplasmic reticulum of the hepatocytes. Once it arrives in the liver, the bilirubin will be acted by the enzyme. Then it reacted to the enzyme to the transferase. This enzyme transfers to glucuronic acid to each of the two propionic acid side chains of the bilirubin, and it result bilirubin diglucuronide. So lastly, this is a acid chain of the bilirubin and will resort to bilirubin diglucuronide. This is now called conjugated bilirubin. Therefore, this is now water soluble. It will be secreted into the bile canaliculi and will join the bile and secreted in the intestine. Since it is into a bile in the colon, it will be acted by the enzyme produced by the enteric of intestinal bacteria. It will now convert the B2 B2 to mesobilirubinogen. With the same enzyme, it will be converted in urobilinogen and will undergo the process of oxidation and become stercobilinogen. Further oxidation will convert in the stercobilin, which is excreted as stool and gives yellow to brownish color in our stool.